So again, I'd like to thank everyone for taking the time to join us today. As Martin discussed, we will introduce you to the M-Series system. We'll talk a little bit about MRI as an imaging modality, just to briefly introduce you, those of you who are less familiar with MRI, although I can see from the poll results there's a number of you who actually do use MRI in your research currently. We'll spend a little bit of time comparing the M-Series systems to conventional high-field MRI systems to help address some of the questions that you may have there. And then I'll spend the last about half of the presentation just discussing different key research applications and showing you some images. So bear with me as we get through the first 2 points. Rest assured that we will spend a significant amount of time on looking at different images and how the system has been used across a wide variety of applications as we go through. So like I mentioned, I'll introduce MRI as an imaging modality to those of you who are a little bit less familiar, and then we'll jump right into the design of the M-Series system and then specifically focus on some of the components of that system, including the animal handling system, the software interface, and the user experience. I'll talk a little bit about some of the hardware and software add-ons that come with the system, expanding its capabilities, and I will spend one slide talking about the purchase price and operating costs of the system. Sorry, bear with me. So for those of you less familiar with MRI or magnetic resonance imaging, you'll see some typical images that we would obtain from the clinic. So these aren't human samples or human patients across the bottom. And so as many of you may know, MRI is considered to be the gold standard for soft tissue imaging. And what does that mean? That means that we're able to differentiate various organs, their internal structures, and tissue types. So across the bottom, you can see images from the brain, the abdomen looking at the liver and the kidneys, as well as pathology looking at a tumor within the brain on the right-hand side. So again, we're able to visualize different tissues, structures within them, and pathological changes that may be happening. Contrast agents can also be used, and that's going to help us to visualize different vascular structures as well as specific molecular targets. Important to note when talking about MRI imaging is that it does not require the use of ionizing radiation, making it ideal for longitudinal studies, especially for the preclinical world. So let's talk about MRI versus other imaging modalities in case you have a background or are currently using other imaging modalities, as many of you are within your research. So MRI and ultrasound, like we talked about before, don't require ionizing radiation, making them preferable for longitudinal studies when compared to things like CT and PET, which both require some type of radiation. MRI is the best choice for soft tissue imaging, including the brain and looking for tumors and different pathologies. Ultrasound is also good for looking at soft tissues, however, it does not penetrate bone and it can't penetrate through air. So this may limit its applications to some of the abdominal organs and the heart, for example. Also with ultrasound, it may suffer from penetration depth issues, especially when looking at high frequency ultrasound systems for small animal work. So that's something to consider when looking at both imaging modalities. CT, on the other hand, does require the use of ionizing radiation. However, it is ideal when looking at bones, structures filled with air such as the sinuses and lungs, as well as kidney stones. However, it doesn't resolve soft tissue quite as well as MRI. PET is slightly different in that it requires a radioactive tracer to be injected into the imaging subject. Often, the tracer used can be FDG, which is simply a radioactive derivative of glucose, and that's used to look at tissues which are highly metabolic. With PET, you should be co-registering those images with some type of anatomical image to provide you context for where that signal is coming from, and we'll look at an example in a second. So again, these images are coming from the clinic, not from the M-Series systems yet, but what we're looking at here is a comparison between CT images and MRI images. And on the CT image of the brain and the spinal cord, you can begin to see that the bones show up very nicely, but being able to resolve the differences within the soft tissue within the abdomen, for example, on the CT image or within the brain, becomes more challenging. When we start to look at MRI images, again, of both structures, we can see very nicely how easy it is to differentiate these different soft tissues. With the MR images on top, you'll see that there's different labels given to them. I'm not going to spend too much time explaining these. We will mostly focus throughout this presentation on T1 and T2 weighted images, and I'll explain as we jump into the applications what this really means and what the different structures are that we're looking at. But know that within MR there's different pulse sequences which allow us to highlight different structures, different features of the tissue. And so that's what the FLAIR and the GRE and the PD come down to. But again, we won't focus too much on that, but I will explain T1 and T2 as we get a little bit further on in the presentation. Here we're looking at a PET image in the center. And so again, this is going to be a heat map of where metabolic activity would be because we're looking at FDG, again, a derivative of glucose. You can see from this image that there's really no anatomical information available, so it should be co-registered with another imaging modality. Here specifically, it's co-registered with MRI. And in this case, we're able to then figure out what anatomical structures are providing us with these very strong PET signals. And I'll show you a few examples from the M-series system later on in the presentation. When we're talking about imaging, we're often considering resolution and the ability to resolve anatomical structures, as well as taking a look at sensitivity and molecular imaging. And when we talk about MRI, we have a really nice combination of high resolution images providing very strong anatomical definition. And when we add in contrast agents, we further increase the sensitivity and molecular information that we're able to obtain from these images. So moving a little bit away from the introduction to MR, let's talk more specifically about these M-series systems. They are a compact magnet designed specifically for use by preclinical researchers. And we'll go into a lot of details, but an overarching theme to this presentation will be that they provide powerful results without the cost, complexity, and technical burden that you may have associated with conventional MRI systems. What you see here is a picture of the M-series. And the M-series is simply a set of magnets connected to the same electronics and the same hardware, and merely the difference is going to be the magnet that you see here. The difference between the magnets, they both operate at 1 Tesla. The difference is the size of the bore or the hole that the animal is going to sit within. The system on the right is the M3, and the system on the left is the M7, as you see on the side. This table just explains some of the technical attributes of those systems. The main thing that I want you to focus on here is that the M3 has been designed mostly for mice, small and large mice, and the M7 can be used on mice all the way up to large rats. And again, that just has to do with the opening of the chamber or the bore, as you see noted here. And just a couple of quick images. We'll get more in detail here, but imaging applications range from neurology through to anatomical or cancer imaging within the abdomen, and looking at muscles or different structures within the hind limbs and thoracic cavity as well. These systems require no special infrastructure to be installed. So you can see on the right-hand side a system placed within a laboratory environment. The system is compact in its design, and very special to these systems is that they are self-shielded. And what that means is that there's minimal external fringe field. So with a conventional system, you'll see some pictures later, you may be used to having the magnet room and you have a Faraday cage or basically the ability to isolate that magnetic field. All of that exists within this compact magnet. And so when we talk about fringe field, we often talk about the 5 Gauss line, and you can see this red line marked on the floor. What that designates is the 5 Gauss line. And you wouldn't want someone with a sensitive medical piece of equipment operating within that, such as a pacemaker, but outside of that, someone with a pacemaker could be within the room working on the workstation, for example. As you see in the image to the left-hand side, electronic equipment can be placed within that 5 Gauss line, even directly on top of the magnet without being interfered with. So there's really very little external fringe field from the magnet. The gradients, which is the way that we acquire the image, and if you've ever had an MRI yourself, that's the noise that you're hearing during your MRI, and so it can be quite loud on a conventional MRI system. The system has been designed to operate very quietly, and that comes into effect in 2 ways. So in the surrounding laboratory environment, people aren't interrupted in the work that they're doing. The animal being imaged is also not disrupted, although under anesthesia, it can be a light anesthesia because there's nothing going on with the animal, so they may hear the sounds and respond. As well, due to the lack of infrastructure, these systems can be placed behind a barrier within an animal facility. And so when they're in that location, we want to consider the effect of any sound created by the equipment on the entire colony. And again, they're quite quiet in their operation, so they can be placed behind a barrier within an animal facility. So as I mentioned, the systems can be installed within the animal facility, within an existing laboratory, right next to other equipment or furnishings. So there's really flexibility in where you're going to position the system within your existing laboratory environment. The systems are typically installed within one day, with images being acquired on that day, and you can start your imaging sessions immediately after. So we'll take a second just to review the different components of the system. So as I talked about a few slides ago, the difference between the M3 and the M7 is simply the magnet. So that would just be switched out here. Remaining the same between all of the systems is the electronics cabinet, which you can see in the far right-hand side here, as well as the workstation and the software interface. So again, those remain consistent between the systems. Included with the systems is an animal handling system, and we'll dive a little bit deeper into that. Anesthesia, which delivers and scavenges any excess gas from within the imaging chamber. And then with each system, you have the ability to add several features on. So a PET insert to allow for simultaneous PET MRI, the VivoFuse option, which allows for co-registration of 3D optical images. So those would be acquired on a system which you already have within your imaging facility, co-registering those with an MR image. And MR-based histology. This allows for the acquisition of very high resolution ex vivo images. And I'll expand a little bit on each of these as we move through the presentation. And although we talk about the system being easy to use, designed for the preclinical researcher, it is also a fully capable MRI system. So you have the ability to program your own sequences if you have that background. As well, you can add on the capability of multi-nuclear imaging. And for those advanced users, that would allow them to image C13, fluoride, contrast agents, as well as xenon-129 and other hyperpolarized gases. So again, I won't focus on those today, but if those are applications that you're interested in, feel free to reach out and we can help provide you with some more information there. Moving to the animal handling system, you can see it up here in the left-hand corner. There's beds that have been designed for both mice and rats. Obviously, depending on the size of the animal, you'll choose the appropriate bed. There's also coils designed for different anatomy, and on the next slide, I'll show you the variety of coils designed, and we'll talk a little bit more about why you would choose which coil. Those do include automatic tuning, so it makes the user experience much easier. The beds do have heated water flowing through them, as well as allowing for physiological monitoring. And like I talked about before, they have delivery and scavenging of anesthesia. On the front of the system, you'll see a manifold, which allows you to change these animal handling systems dependent on the coil or the size of the animal that you're working with quite quickly, just by simply connecting and reconnecting these connections here. So here we have a list of the different imaging coils. So again, we have beds which are sized appropriately for mice as well as rats. And we have imaging coils for the head, body, or even larger animals like large mice and large rats. And you want to choose the coil that fits most appropriately to the imaging target. And when we talk about comparing high field and low field systems, we'll talk about the importance of finding the coil that fits as tightly to the anatomical structure as possible. That's going to give you the highest signal to noise and therefore the best image during your acquisition. We talked about heating water going through, so we include a circulating water bath, and that's going to allow the heated water to circulate underneath the animal in the animal handling bed to help maintain its core body temperature throughout your imaging session. You would then want to be able to monitor different physiological parameters as well. With the system, you can look at ECG, heart rate, respiration rate, as well as body temperature throughout the imaging. You can choose which of these you'd like to record. Typically, we look mostly at respiration, but you can include temperature. And when needed, ECG can be acquired as well, and that would allow for respiratory and ECG triggering of your data if required. Anesthesia is integrated and included with the system. It's an isoflurane-based system that also includes scavenging. The scavenging will help protect the operator from any excess gas which is not used by the animal. It also helps to protect the animal during the imaging session to ensure that isoflurane doesn't build up within the imaging chamber, causing over-anesthetization of your animal. The vaporizer has 3 delivery points. The first one is typically used for the knockdown chamber. They are individually controlled, so you can choose which ones are on or off at any point in time. The second one would go to the animal bed to maintain anesthesia throughout your imaging session. And the third one is there for any animal preparation steps. For example, if you're using contrast agents and you need to place a tail vein, you may want to be doing that off on the side while your previous imaging session is being finished up. So again, individually controlled and 3 access points there. Let's take a minute just to look at a workflow video. This will help you better visualize how you would prepare the animals for imaging and what's really involved. So the first step is to anesthetize the animal. Again, they're going to go into the knockdown chamber and isoflurane will be delivered. Once the animal is asleep, you'd hook its teeth onto the bite bar. The anesthesia is also delivered through that white area. There's 2 holes there that would deliver the anesthesia. The nose is pulled into the mask, and that's where the active scavenging would come into play. If you notice the blue pad underneath the animal, that's for respiratory monitoring. And as the animal breathes, they would simply press onto that pad and that signal can be monitored. The coil is then placed over top of the animal with the anatomical target positioned within the middle of that coil. The head coil is actually integrated into the anesthesia mask, so in that case you would simply pull the animal's head into the coil at the same time as you're pulling it into the anesthesia mask. Once the animal is in position, it's simply placed inside the magnet, and everything else is done within the software. So the software has been designed to be easy to operate and intuitive, and it's also designed for preclinical studies. And what that means is that you can have information about the study, the animal identification, and all of that is saved with your images. This allows you to quickly generate reproducible and quantitative results. As we've mentioned throughout the presentation today, there really is no need to have a background in MR physics. And why is this possible? Well, the system comes with a number of default sequences which have been optimized for use with a number of different application areas. And so basically you would go through, you would identify the sequence that you want to operate, choose the number of slices and where they would be located within your animal, and you simply run them. So for those who have a basic understanding of MRI, you don't have to work with the different pulse sequence parameters, but the system does offer those who are more familiar with MRI the ability to customize those pulse sequence programs, optimize them to meet your specific needs. The system does also allow for sequence programming itself, if that's something that you want to delve into. And as I mentioned earlier, we're going to kind of stray away from that in this presentation, but if that's something you're interested in, feel free to reach out and we can provide you with more information. This is what the software interface looks like. On the left-hand side, you can see the different imaging sessions which have gone on. You are able to enter the ID of the animal. You would put the orientation of the animal so that the labels on the image are appropriate. Select the coil that you're going to be using for that session. And you also have the ability to load a protocol set. And what that means is you go through your studies, you always want to be acquiring the same images on every animal at the different time points that you're looking. And you can pre-save a set with all the same parameters, and you would simply, again, make sure that the slices were in the right location and simply run that entire set. And that's going to save you a tremendous amount of time because it will go from sequence to sequence to sequence, taking images in order as you prepare for the next imaging subject to be ready. And you can see a protocol set here on the left-hand side as an example. Information about that animal can be entered, including its gender, date of birth, and there's a comment section for you to enter any specific information to do with that animal. So again, that information can be retrieved as you're doing your data analysis later on. Moving now to some of the add-ons with the system. So the first one would be the PET insert, so the SimPET. This expands the capabilities of the M7 system specifically to allow for simultaneous PET MR imaging to be acquired. So you can see the animal handling system that we talked about before going in on the right-hand side of this magnet, and the PET insert would go on the other side. And again, those images can be obtained simultaneously. The PET insert is highly sensitive and is able to detect functional information, looking for abnormalities and early disease. As well, the MR is then going to obviously provide you the anatomical context to go with those PET images. The VivoFuse, as we talked about earlier, is a hardware and software add-on. And what this does is it allows you to acquire a three-dimensional tomographic image on one of your optical systems, for example, the IVA spectrum, and then take the animal, transport it to the MR, and acquire a 3D image. By combining those images, we get both the molecular and functional information from the optical image, along with the anatomical information provided by the MR. And what that can allow you to do in a complex tumor model that you're seeing above is that you confirm the presence of a tumor using the optical image, and you are able to then quantify the tumor volume using the MR image. The final add-on that we'll talk about today is MR-based histology. The goal of histology, MR-based histology specifically, is to acquire a very high-resolution 3D image of a fixed ex vivo sample. So where this comes into play a lot is with toxicology studies. Here, the software allows for automated sample handling so that very long images can be acquired to get those high-resolution images. But we have automatic sample handling so that, for example, overnight you could run several samples through without the intervention of a user present there throughout the night. The images are then used to detect, localize, and quantify the number of lesions within the sample. As I promised at the beginning, we will spend one slide here talking about the costs of the system. So first off, a well-equipped M3 system, which again is for mice, is around $290,000 US. The M7 system, again going for rats, is closer to $400,000 as a basic cost of the system. But we all know with MR, there's many other costs that are associated with those. I'll focus on the installation a little bit later when we're comparing with conventional MRI systems. But we also need to talk about running costs and ongoing maintenance costs. So with the design of the system, we talked about it being a compact permanent magnet. Being a permanent magnet, that means that there's no electricity that's required to create the magnetic field. So there's no cost associated with creating the magnetic field. Additionally, there's no heat being generated to create the magnetic field. And that's where the cryogens or the water come into play with other conventional MRI systems, is that the heat that's required to create the magnetic field has to be dissipated or the system would overheat. In high field system, that often requires helium. And in some other systems, they may use water cooling. Both of those cost a tremendous amount of money to maintain, as well as the helium. Prices are going up around the world these days. With the M-series system, the only electricity that's required is to run the electronics, and that's only going to be on during your acquisition, and that takes standard electrical signals, so there's no excess cost associated with operating the system there. As well, there's no ongoing maintenance costs with the system. When we talk about a conventional MR system, maintenance contracts may be in the order of $100,000 to $125,000 per year, and that doesn't even include any repairs. So again, those costs are minimized, if not completely removed, using the M-series system. So what does all of this mean? It now means that MRI is more accessible for you as the preclinical researcher, and that your budgets can be preserved for doing the science that you want to do rather than purchasing and maintaining equipment. So moving now on to the comparison of the M-series system to a conventional high field system. So we'll start with looking at some image comparisons between high field and low field systems. We'll talk about contrast imaging at 1 Tesla, again, the operating field of the compact M-series system. And we'll talk a little bit about installation requirements, and I'll show you some images from installs for both systems. So when we talk about field strength and signal to noise, the laws of physics explain that signal to noise ratio is linearly proportional to field strength. So what that means is that 1 Tesla compared to 7 Tesla, you will always have a higher signal to noise, but how can that be improved and what does that mean to the work that you're doing? So the images that you're seeing here, we can see the kidney, for example, at 1 Tesla and at 7 Tesla. And the question that I want you to ask really is, are the images of the M series enough to answer the biological question that you're being asked? So here, if you're looking at the size of the kidney, or if this was a tumor, for example, would you be able to accurately outline the tumor volume of this image? And I think the answer here is yes. But of course, how can we improve signal to noise? And with the M series system, a lot has been done to optimize the image acquisition to get a better signal to noise ratio. So we can do that in multiple ways. We'll talk next slide about the acquisition time and resolution. But the coil design and the electronics are very important as well in this. And we talked earlier about picking the coil which fits as tightly to the imaging subject as possible. So having those body and head coils becomes extremely important, and you would want to use the one that's most appropriate for your imaging application. When we talk about resolution and acquisition time, again, we need to ask ourselves, are the images sufficient to answer the biological question that we're asking? So here we're looking at resolution of 156 microns compared to 312 microns with increasing acquisition time, obviously with increased resolution. And again, if we're looking at the images here on the right-hand side and we're looking at the kidney, in both images, I feel like I'm able to visualize the kidney well and could measure its volume quite easily. So again, is the image with a resolution of 312 microns taken at 5.5 minutes sufficient to answer the question that I'm asking rather than taking a longer acquisition time? And I think here you would agree the answer is yes. I mentioned earlier that I would explain a little bit more about T1 and T2 weighted images. And really what this is doing is highlighting different contrasts within the image. When we're looking at MRI, we're looking at protons and how they are distributed throughout the body. And so you can imagine there's protons in water, fat, and lots of other structures within the body. So on a T1 weighted image, the fat is going to appear bright. And actually on the images over on the right-hand side, we can see that very nicely in the fat pad sitting below the kidney. The fat will appear bright and the water will appear dark. The water content is higher in the medulla of the kidney compared to the cortex. And like we would expect, the inside of the kidney is going to look darker. On a T2 weighted image, the exact opposite is going to be true. Water is going to become bright, as we see again in the medulla, and fat should become dark. Specifically, the way that these images are acquired, they're a mix of T1 and T2, so it's called a fast spin echo, and that's actually going to make the fat look bright. However, what we want to concentrate on here is that the water becomes bright on a T2 weighted image. And we'll see some examples where in inflammation and different pathology, that becomes very important. It is what allows us to visualize the pathology in those images. Jumping back to high field and low field systems again for a second, some tissues respond differently at 1 Tesla compared to high field systems. This specific example from a glioblastoma, no contrast agent was used here, but what we can see is that the glioblastoma in the brain shows up very brightly, and the borders are more clearly defined at 1 Tesla than you would see at 7 Tesla. The structures that you're seeing next to the tumor, these bright areas here, these again are a T2 weighted image, so that's going to be water. That's actually the cerebrospinal fluid within the ventricles of the brain. So we'll see that as we go through in different neurology applications. When we talk about contrast agents, there's two different types of MRI contrast agents, gadolinium-based contrast agents and iron oxide nanoparticles. Those are the two main ones. There are some others, but we'll talk mainly about those two. Gadolinium-based contrast agents actually respond better at 1 Tesla than 7 Tesla. And this was a paper published by Silvio Amy and his team in Torino, Italy, where they took a look at the sensitivity of the systems to gadolinium-based contrast agents, both in vitro and ex vivo, or in vitro and in vivo. So here, at different concentrations along the x-axis, we can see the signal enhancement along the y-axis. And at all concentrations, the 1 Tesla system showed increased sensitivity or signal enhancement to the contrast agent. And if you look here at the lowest concentration, when operating at 7 Tesla, I would not be confident in detecting any signal enhancement that was statistically significant using this level of contrast agent. However, at 1 Tesla, I think it's clear here that the signal enhancement is enough to be sensitive to that signal. When working in vivo, again, it's 7 Tesla on the left, 1 Tesla on the right. Here we have a subcutaneous tumor before and after contrast was injected. Before contrast was injected, we can visualize the tumor. After the contrast, when we look at 7 Tesla, there's very little signal enhancement using that contrast agent. However, at 1 Tesla, we can see a very strong response within the tumor, and the numerical quantification obviously goes along with that. So again, using contrast agents, specifically gadolinium contrast agents, you're going to have increased sensitivity to those agents when operating at 1 Tesla. Some imaging comparisons, this time from the group at Texas Children's. This group actually has access to, I believe it's a 9.4 Tesla system, as well as several M-series systems. And here, using again a gadolinium contrast agent, they're able to look at the very exquisite detail here of the cerebrovascular structures using their contrast agent. Finishing up our conversation about comparing conventional high field systems with the M-series system is talking about installation. And so here you can see a high field system being installed within an institution. And this often requires a crane to lift the high field magnet into position. It can at times require opening up of walls to be able to fit the system into an existing facility, or you would plan the placement of an MR system during the building of a facility. So you would place it prior to any structures being built around it. Here you can see a suggested configuration of the system. So a high field system would have its own room. And again, that's going to have a Faraday cage or a metallic lining within the walls to help shield the magnetic field from coming outside of there. In a separate room, you're going to have all of the electronics which help to operate that system, as well as the cooling devices. And then you're going to have a separate control room where the operator can work at the console to acquire those images. So again, there's quite a large investment here in terms of infrastructure costs in placing the magnet once it's been purchased. For the M-series systems, like we talked about before, they are actually wheeled into position. So you can see the cart actually has wheels. The magnet's placed onto the cart, and it's simply wheeled into position along with the electronics cabinet. And again, there's no infrastructure required for shielding because that's all done within the magnet itself. And the systems can be placed, like I mentioned earlier, within an animal facility or next to other laboratory equipment or fixtures. So right now I'd like to take a second to launch our second poll.
All right, thanks, Tonya. This is Martin Hess again. I'm going to just jump in here for a moment. There's been a lot of great information shared, and before we get into the last part of our presentation, where we provide some examples of research applications and share some data with you, we'll give Tonya a moment to catch her breath. We'll also provide you an opportunity just to take a moment's break. And if you could answer the following question: do you currently have access to an MRI for your studies? And there are 4 options to choose from here. So yes, you do, and you're satisfied with the costs associated with conducting your studies. The second option is yes, you do have access to MRI, but it might be expensive, or perhaps you're having a difficult time booking time with that system. The third option is no, but you'd like to use MRI in your work. And then the last option is that you don't have a system and you don't feel there's a need for MRI in your study. So take a moment to complete this poll, and then we'll have Tonya finish up the second half of her presentation.
Excellent. Thanks everyone for taking the time to respond to that, and Martin for allowing a quick break there. So I'll end the poll and we'll move on. And so like I mentioned in the beginning, I do want to spend the last half of the presentation taking a look at key research applications. So we'll take a look at some anatomy and morphology. We'll talk about some neurology applications, cancer, cardiovascular. We'll touch back on some multimodal imaging and some ex vivo imaging. So jumping right into it, we've talked a little bit about the T1 and the T2 weighted images, and here's just a look at different anatomy within the mouse. So again, we've talked about the different structures within the kidney, looking at the cortex and the medulla and how different they look, both on the T1 and the T2 weighted images. We can begin to look at the liver and the stomach and intestines, as well as the muscles within the hind limb, as well as some fat structures as well. And we'll talk about some metabolic disease models as well. Of course, when you acquire an image, you want to be able to make some type of quantification. And so included with the software is VivoQuant software, which allows you to both visualize your images. You can do some pre and post processing and co-registration of images with other imaging modalities if you'd like. The tool also includes some segmentation tools, which would allow you to generate volumes. These can be done completely manually, semi-automatically, or completely automatically. It just depends on the images and the tools that you're working with. Taking a look at inflammation. So we introduced the idea that on a T2 weighted image, water is going to look bright. So if we induce inflammation here by a topical application of an irritant, we're going to increase swelling and blood flow to that area. And what that's going to look like, again, on the T2 weighted image, is a bright region compared to the surrounding muscle. And here, we're able to quantify the volume of that lesion, the overall leg volume on that side, as well as the contralateral side shown in green. Here we're looking at fat segmentation, so this may become important in metabolic research. So here we're looking at a T1 weighted image where fat is going to appear bright. And using that VivoQuant software, we can place a seed point and we can say connect any voxel which is connected to this one, which has a similar intensity. And in that way, it allows the segmentation of the fat to be much easier than manually segmenting out the different fat pads and regions of fat within this animal model. Taking a look now at neurology, this is a normal mouse, and we're able to step through the brain in whichever orientation the images are acquired, or other orientations depending on how we want to visualize the different structures. And what you're able to see that on this T2 weighted image, we're able to see the different structures within the hindbrain. We can see the cortex and different structures. And again, I mentioned before, the bright areas that we see here are going to be the cerebrospinal fluid within the ventricles. And where that becomes important is when we have disease, we may see that these aren't draining or flowing quite as well, and so they're going to become enlarged, and that's going to be immediately obvious, and you'll see a few examples of that as we go through. So here we're looking at an orthotopic glioblastoma tumor model. The image that you see now is at day 4, post-injection of the contrast of the tumor cells. And right now we don't see any abnormalities, we don't see any evidence of the tumor at this point. But of course, we want to follow that same animal longitudinally. So 11 days later, 15 days post-injection, we are able to see the tumors beginning to grow within the brain. We're able to see the buildup of fluid within the ventricles very clearly here. And this group had a history of injecting the tumor cells within the ventricles themselves rather than sticking within the brain tissue. And so we'd often see that the tumors would spread, as you see, throughout the brain. And so we're able to identify each portion of the tumor as it does spread throughout the brain and follow again that same tumor longitudinally. Looking at an example of traumatic brain injury, or TBI, here a percussion injury was induced on the skull. And again, this is going to cause inflammation. So on a T2 weighted image, we're going to see a bright spot form, and we're able to easily visualize that and quantify its size. And therefore, we're able to take a look at different preventive measures or therapeutic interventions to see how that affects the size of the lesion that's formed as a result of this injury. Here we're looking at a model of epilepsy. So in this case, the animal was exposed to peroxone, and this causes significant symptoms to develop. 3 hours after the exposure, we can see that there's very little changes within the brain. These images were taken on a rat. At 48 hours after, we can see significant changes occurring within the cortex of the brain, and we see those as a bright structures forming, so again, indicating some type of inflammation within that region of the brain. And I'll just let these images step through again so you can see them. Moving on to a stroke model. Here we have a stroke induced by photothrombosis, but similar images, although a larger lesion, could be caused by middle cerebral artery occlusion. So again, a lesion is going to look bright on the image, so the borders can clearly be defined. And we can follow that same lesion as it develops and resolves over time, or monitor any intervention that we want to apply to that animal to try to resolve or protect from the stroke lesion being formed. Moving on to cancer biology. So we can take a look at all different kinds of tumors. Here we're looking at a xenograft of a head and neck tumor located on the hind limb. And so we're able to obviously identify the tumor, and we can look at structures that form within it. So here we're seeing a bright area. This is a T2 weighted image. So that would indicate something that's more fluid-like. So it could be a cyst forming within the tumor, or it could be a necrotic core. And of course, as we're able to define the edges of the tumor, we would be able to quantify the tumor volume. Once we're able to identify and quantify our tumors, we obviously want to be able to see their progression over time. So here we're looking at 2 animals from day 10 to day 23, and we can watch the same tumor as it grows over time or responds to therapy over time. And in this way, we're adding power to the data that we're generating because we're looking at that same animal over the course of the longitudinal study. And as I mentioned before, we of course want to start to take a look at therapeutic interventions. So here we're looking at an orthotopic cervical tumor, and we're able to identify the tumor and look at its response to treatment over time. This is a really great example of where it's always very important to identify normal structures before you begin to identify abnormal structures. And in the cervical tumor model, it was very important to identify the bladder before we identified the tumor. And so on a T2 weighted image, if you remember, the water is going to be very bright, so we were able to clearly identify the bladder, and we know that the cervical tumor should be located very close to that, and so we can identify this round structure as being the tumor. Taking a look at a few cardiovascular images. So MR can also be used to look at cine loop images, which is going to allow us to get a really nice image of the cardiac structures as they move through an entire cardiac cycle. And here you can see that both short axis and long axis images can be acquired, and we can see the blood as it circulates throughout the heart. Likewise, we can also begin to look at the structures that surround the heart and see if there's any changes going on there as well. Of course, we have images, we want to quantify them. So there's different tools that can be used to quantify cardiac function, either again in the long axis or short axis. Here we want to outline the endocardium and the epicardium, and we'll get measurements of ejection fraction, volume measurements such as stroke volume and diastolic and systolic volumes, allowing us to look also at the wall thickness and LV mass. Moving back to some multimodal imaging. So here looking at PET MRI. In this situation, we're looking at a metabolic metabolic research, wanting to study brown fat. And so on MRI, both brown fat and white fat show up bright. There's no differentiation there. But as some of you may know, brown fat is more metabolically active than white fat and can be very important in looking at different metabolic diseases. And so here, we use FDG PET. So again, the glucose derivative allows us to identify metabolically active structures. And so when we identify a hotspot and co-register it with the MR signal, we're able to identify that what we know is fat from MRI is actually metabolically active, making it brown fat, and we're able to quantify the size of those fat pads within these animals. Taking a look at multimodal imaging as it corresponds to cancer research, here we're looking at a tumor. And again, tumors are often metabolically active, so can be identified using PET. And here we can see on the PET image that there's an area on the outside of the tumor which is quite active, but on the inside of the tumor, there's really no PET signal. And when we co-register that with the MR image, we can see that that area with very little PET signal corresponds to a bright area, which is most likely a necrotic core within that tumor. And of course, with the VivoQuant software, we can add in any other imaging modalities such as CT to have a very nicely co-registered images with all those imaging modalities. Talking about the VivoFuse add-on that we discussed earlier, the hardware component of it is actually this cassette. And what that does is it allows you to position the animal and maintain its position as you move from the optical imaging system into the MR. And by doing so, you're able to exactly co-register those images because the animal has not moved between the acquisition within your optical imaging system to your MR image. And by co-registering those images, as we talked about before, you get the power from the molecular and functional information from the optical co-registered with the anatomical image provided by the MR. And again, we can quantify the lesion size and tumor volume, for example, from that MR image. Finishing up by looking at some of the ex vivo imaging applications. So again, to remind you, the purpose of MR-based histology is to acquire very high resolution images on fixed samples. So here we're looking at a whole rat brain that was scanned with very high resolution. We're looking at 83 microns in plane resolution. And with that, you can see the exquisite detail of this brain and any pathological changes that were caused by any intervention or a disease model that may have existed within this animal. We can also start to look at fibrosis, for example. So here, a control versus fibrotic liver, and you can see the very strong difference on MR. The MR is a non-destructive way of imaging the sample. So the sample can then be sectioned for conventional histology. And for example, Mason's trichrome stain can be performed in which the blue signal that you see here is the fibrotic tissue, which corresponds very closely with the MR image that you see here. Where MR-based histology becomes really important is with toxicology studies. Conventionally, only a few slices are taken from an organ to look for lesions to perform the toxicology studies. So for example, if there was very few lesions within the liver and your histological sections happen to miss all of them, then you may not be accurately assessing the toxicology of the substance that you're looking at. However, if you can do MR-based histology before performing conventional histology, you get a full 3D image of the liver, for example, as you see here, and you're able to identify lesions throughout the entire organ, and then you can use the placement of those lesions to guide your histological sectioning, that you can then confirm what those lesions are based on conventional histology, as you see here. So at this point, I'd like to take another quick break and run our third poll.
Thanks, Tonya. We'll run this last poll. Tonya will wrap up, and then we'll do a short Q&A. Just a reminder to everybody, please do feel free to submit some questions for Tonya through the Q&A chat window. We have one or two already. We'd like to see more. So the last question we'd like to ask you, based on some of the information Tonya's just provided, would you be interested in learning more about a specific add-on? And the options are the SimPET, simultaneous PET MRI add-on, VivoFuse for co-registering optical and MRI images, MR histology for ex vivo imaging, and if there's a specific imaging application, please do specify in the chat window. So I'll let you respond to this poll, and Tonya will take over.
Wonderful. Thanks everyone for taking the time to answer that last poll question that we have. So just to finish up, I'd like to just run through what we talked about today. So we introduced you to the M-series system. We gave you a little bit of background into MRI, so I hope that was helpful for those of you who aren't currently using it in your research and aren't familiar with it. We talked about the features and the benefits of the MRI system, and as Martin introduced in the beginning, it really is a cost-effective solution that allows the preclinical researcher to access this imaging modality, which in my previous lives had always kind of been held up on a pedestal of, MRI is great, it resolves soft tissue, but it's not attainable for someone who doesn't have the background in MR physics. And these systems really do allow those who don't have that background to harness that power and use it within their research. We talked a little bit about comparing a conventional system to the M-series system. And then we focused a lot of time on looking at different key research applications. And we hope in the future to run a number of different webinars taking a look at different papers which have been published in specific areas. So you can, again, start to see how your peers are using the system in their specific research applications. And I just hope that you can leave today imagining how the system could help you in the research that you're doing to get more powerful and strong data from your imaging models that you're working with now. Right now, I'll turn it back over to Martin, and we'll start with some question and answers for the last couple of minutes.
All right, thank you, Tonya, and we'll try and finish on time. We might just run a few minutes late, but a reminder, we'll be summarizing all our Q&A responses for you, and we'll distribute after the fact. We had some questions come in before our webinar started today. The first one is, what are the advantages of using MRI compared to other imaging modalities, such as ultrasound and optical imaging?
Sure, it's a great question. And so, as I talked about in the beginning, MR has always been considered to be that gold standard for soft tissue imaging. And so when comparing it to looking at optical or ultrasound images, we want to be able to look at penetration depth, for example. So MR is able to see completely through the different tissues, which, as I mentioned, ultrasound, when working at the high frequency sounds that are often used for small animal images, it may suffer from some depth of penetration issues. As well, ultrasound can't see through the skull, so it can't be used for neurological imaging in most situations, or into the bones or lungs. MR, on the other hand, can see through all of those and has no issues being able to see tumors, for example, within the lungs or structures within the brain. Compared to optical imaging, MRI provides anatomical structures and information that optical doesn't provide, but again, with that VivoFuse option, we can co-register those images together to provide the anatomical context for that optical signal.
Excellent. Thank you very much for that answer. And you may have mentioned this already, but what is the field strength of the system?
Definitely. So I mentioned it a little bit throughout. The M-series systems both operate at 1 Tesla. And if you recall from the earlier slides, where that becomes important is talking about the quality of the images and the signal to noise, and understanding that these systems have been optimized such that the electronics, the pulse sequences, and the coil design all start to overcome any limitations due to the field strength. I did see one question come in regarding, what are the disadvantages of operating at low field strength, and that is an important part to talk about. And the question specifically came around diffusion tensor imaging and functional MRI. And of course, when you're looking at very specific applications like those, they do require high field systems. The M-series is capable of doing diffusion weighted imaging, but diffusion tensor and functional MRI has to be done at high field systems. And we would recommend that you use those systems because you want the tool that best helps you answer the biological question that you're asking.
Perfect. Thanks for that great answer. And hopefully that does answer Dr. Zheng's question. If we can certainly elaborate on that offline if we'd like to provide more information.
Definitely.
Perhaps a difficult question, but what is the resolution of these systems, Tonya?
Yeah, absolutely. And this question did come in beforehand, so I actually brought back a slide that I talked about in the beginning. And so the answer to the question, and I hate the answer of it depends. So with MR, there's so many things that go into the resolution of an image. And so really we can get images we saw as low as 83 microns. They can be higher resolution with longer acquisition times. So it's always that balance of resolution and acquisition time. So again, we see 2 examples here of 156 microns and 312 microns and the acquisition times associated with those. So again, you just have to choose the resolution which helps you to answer the biological question that you have, of course, optimizing the acquisition time such that your imaging sessions aren't getting too lengthy for the studies that you need to be conducting.
Okay. And again, you may have mentioned this in your presentation, but how were the volumes quantified that you showed throughout your presentation?
Great question. So again, the systems come with VivoQuant software, and what that does is allows you to do all the post-processing offline. And the benefit of having a separate software package to do the analysis afterwards is that the time on the system is maintained for acquiring images. Now, within the acquisition software, there are basic tools for visualization and making some simple measurements so that as you're acquiring the images, you have a sense of what's going on within your animal model, but you would then take those images and put them into the post-processing software tool to allow you to get those full 3D volumes and different visualization tools that are accessible through that software.
Okay, fantastic. We have lots and lots of questions coming through. I don't know, Tonya, if you want to also maybe just scan through the list and see if there's anything we want to address now. We will not be able to get to them all, but we probably have a few minutes if we want to continue on with our Q&A.
I'm just taking a quick read through. Some of these are very specific to the researchers, so we will address those individually as well as make the transcript available for everyone. Dr. Zhang had a follow-up question in terms of what is the format of the images. And so the images can be exported in any number of different formats, definitely with a DICOM format, I believe is the standard way of exporting. And it's a question I can answer more specifically in the transcript. But within VivoQuant and every other imaging analysis software that I've worked with, I've been able to take the images out and look at them within ImageJ, lots of different formats there, so there's no proprietary format used with the system. And a final question asking about the PET insert. The PET insert is unique in that it can be used, it can be inserted into the system, and once inserted, it can be used on its own to acquire just PET images if that's something that you want to do. And if you don't want to acquire PET, then you would simply acquire the MRI image. So you've got full flexibility in terms of what acquisition you want to take during your imaging session, whether it's simultaneous PET MRI or individual images from either modality at that time.
Okay. Perfect. Well, I think at this point in time, Tonya, if you would agree, we've exceeded the 60-minute mark for our session today, and we want to be respectful of everyone's time. So we'll wrap things up. And as mentioned at the start, we will be sure to answer all these outstanding questions privately, and we will distribute a written transcript with all the questions and answers for all of you to see. I'd like to thank Tonya for her presentation today. Thank you very much. And I trust that we've been able to demonstrate how valuable and accessible and user-friendly technology like the M-Series MRI system is, and how it can enhance the quality of your research and increase throughput in your lab. And I would encourage you to reach out to us if you still have questions. We'd love to learn more about your specific application and how we can support your research. So with that said, thanks again to all of you for taking time out of your day to attend our session, and we look forward to seeing you at a future Syntica event, and have a great day.